Hi class, Mr. Roth here. Today's lesson is going to be on the central dogma of biology. So what is the idea of the central dogma? It's basically the idea that it's our DNA that is responsible for what proteins are made and at what time. And so you might be asking yourself the question, well, what does it matter what proteins we have? What does it matter when they're made? Well, it turns out that when and what is made is really what ultimately is determining whether our body is functioning correctly as a human body should, or whether or not it's not functioning correctly. And we've talked about some examples already of what happens if the wrong protein is made at the wrong time, or if a defective protein is made. For one, we know that if a defective repressor protein is made, that we cannot regulate the cell cycle, and at that point we have what we call cancer, which is where cells are dividing uncontrollably. So we know from examples previous in class that proteins being made in the right way and at the right time are crucial to the functioning of cells and ultimately the functioning of our bodies. So over here I would like to draw your attention to this cell. And you'll notice that the cell has a membrane and it's also got a nucleus in the center. The nucleus in the center contains our double helix of DNA, which holds all of the information we need to make those proteins. And it's sort of a two-step process by which we finally get to that protein. And the first step is something we call transcription, and that's what's occurring between here and here. And if you notice, right here we have this double helix of DNA unwound in this particular section right here. It's been kind of separated. And there's a particular enzyme called RNA polymerase that's going to separate this. The RNA polymerase has really two functions. One is to separate and unwind that DNA double helix, and the second is to synthesize this new strand right here. You'll notice if you look closely, there is a new strand being made, and it's being copied off of this bottom strand of DNA. So really, this bottom strand of DNA is the template by which we make this new RNA strand. So if you correspond what's happening here over to this image, you notice that transcription is the process by which we go from DNA to a single-stranded RNA. And that's all occurring right here inside the nucleus. And so the product, just like we have over in this picture, is a single-stranded RNA. Okay, so you might be asking yourself the question, why do we have that middle step? Well, the answer is that we need the DNA to stay in the nucleus, and so we need to make a messenger. And really, RNA is simply a messenger molecule that can leave the nucleus and go about the cell and finally go and make proteins. And so the very next thing that this RNA will do is it will, just as we said, it will leave the nucleus and it will go and find a ribosome. The ribosome will then help to get that final step accomplished, which is what we call translation. Okay, so let's break it down. Again, as we said before, the first step going from DNA to RNA is transcription. I'd like to highlight again that this blue blob here is an enzyme. It ends with ASE, and it's called polymerase, RNA polymerase. And so, as the name suggests, it's going to polymerize an RNA strand, which means that it's going to take single RNA nucleotides, connect them together based on the template strand of RNA that is exposed. And so you'll see coming out of this RNA polymerase is our created RNA strand. Okay, so once this whole process is accomplished, again, we're only going to transcribe a certain segment of this DNA. We're not going to do the entire chromosome. We're just doing a portion. We're doing a gene, as it were. And once the gene has been transcribed, we then have our RNA strand. Okay, so let's just say that we have already synthesized our RNA strand and it is floating around the nucleus. As I said before, the next thing it'll do is it'll snake out one of the pores and go and attach itself to a ribosome. Now these ribosomes can be free floating. They can just be floating around the cytoplasm, but it's very often the case that they're associated with the rough endoplasmic reticulum. What actually gives the endoplasmic reticulum its rough appearance is that it's studded or embedded with these ribosomes. Okay, we're now at the last step of the central dogma, which is called translation. This is the process, if you look at our image up here, by where we go from RNA to proteins. So how does that process occur? Well, as we said before in this image, it needs a ribosome, and there's really three things involved: ribosome, RNA strand, and then our final product, which is our protein. Just to remind you, our protein really is nothing more than a chain of amino acids. Amino acids are the monomers that make up a protein polymer. And so we know the ribosome needs to bring these in and connect them together. And this is what we see. This image gives us a little more detail of the process, but floating around the cytoplasm are things called tRNAs. And these tRNAs they have a particular sequence here that needs to match up with the sequence on the RNA. Whatever the sequence is here, it corresponds to a very particular amino acid. And so the order of amino acids that end up in the chain is ultimately determined by the sequence of nucleotides that are found in the RNA, because we know that they have to match up. In other words, A's always bind to U's in RNA. Similarly, if we find a G, they always go across from C's. So ultimately, what determines the order of these amino acids is the order in the RNA. Okay, once this entire RNA has been translated, the final product will be a polypeptide chain of amino acids, also known as a protein. Once that protein is created, we then have the product. It can go float around the cytoplasm and perform its function. In fact, one thing I would like to point out is this. Sometimes the protein that is being made is in fact a ribosome. So there are actually RNA strands that produce a ribosome as the final product. Kind of a chicken or the egg scenario, which came first. Biologists have pondered that question for years. But anyways, I hope you guys are making sense of this central dogma. We're going to get much more practice in class. Adios.